Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your question. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, John Harmer, Strategic Initiatives Leader, Aseptic Filling from Cytiva.
Thank you for the introduction, Alex. As you mentioned, my name is John Harmer, and I lead strategic initiatives at the Aseptic Filling business of Cytiva, and that was formerly Vanrex Pharma Systems. We were acquired about a year and a half ago, and we're now fully integrated into the Cytiva's businesses. And so what I'll be talking about today is how you can grow your CDMO, your contract manufacturing business, by adding aseptic filling as a service. And I'm going to focus on the use of Gloveless robotic aseptic filling work cells as an example. So when we look at the current issues facing CDMOs, one of the main ones is the fact that these are problems that face not just CDMOs, but also their potential clients or customers, the biotechs that are developing new drugs. And so the pandemic revealed a shortage of capacity for large volume commercial vial filling. And so that was addressed by a fairly significant increase in capacity for those situations. Unfortunately, the new capacity isn't appropriate for clinical trial production. And there's a lot of built up demand or unmet demand for clinical trial products. And so there really is currently insufficient capacity. So many CDMOs either do not offer aseptic filling or only have conventional systems with long changeover times, so the larger systems I was talking about. And the other aspect, of course, is in clinical trials, every customer brings different manufacturing needs. So to serve this large body of customers that'll have a wide variety of products, you need to have flexibility. The other thing is you need to make sure that you can get the drug product quality that the customers expect. Regulatory agencies are expecting a higher degree of quality and greater patient safety. They're always pushing to increase that. And so you want to be able to provide your customers with the appropriate safety assurance. Very often for early phase clinical, the choice is manual filling, but that can compromise patient safety because there's a large risk of operator contamination. It ends up being actually a very cumbersome process with the need to qualify many operators and the environmental monitoring. So there are some issues with manual filling. On the larger end, for later stage clinical production, customers are often forced to go to larger scale systems that are designed for commercial. And those can be hard to transfer to. They're not an efficient use of those larger lines. And if you're installing one of those larger lines, you're only going to be serving a small portion of the market, and the time to install will likely be significantly longer. So when we look at what the solution could be, we're going to look at what we call Gloveless robotic isolator technology from Cytiva. And so there's two systems here, the microcell and the SA25. And, if you use these to expand your manufacturing capacity, you can add it quickly because these are standard systems. You can address the flexibility issue because they fill multiple formats. The SA25 can fill vials, syringes, and cartridges on the same machine with minimal tool changeover. And they all use ready-to-use components in standard configurations. And they're also able to deliver very high quality. You get the highest amount of aseptic assurance, ensuring that the products are sterile. They have no gloves, no place for the operator to interact with the products. And they are simple and standardized, so that reduces risk. They're also at the appropriate scale for production, and I'll go into that. The microcell vial filler is really designed for multiple small batches. The standard batch cycle is about an hour, and it would fill 3 tubs of ready-to-use vials. Those are up to 300 vials. And if you need to do more, you simply repeat that cycle. And they can fill vials, of course, from the 2R size to the 50R size. So a very wide range of vials. So that makes it ideal for small batch production. It can become on-demand production for small batches. Very little setup time, very quick turnover. If you're a contract manufacturer with multiple lines, it can basically take capacity off your large line and allow you to do small batches very quickly and efficiently. The SA25 is perhaps the workhorse of our contract manufacturing customers. It'll do batches anywhere between 1,000 to 20,000 units, so ideal for late stage commercial as well as, late stage clinical as well as niche commercial production. And as I mentioned before, it's vials, syringes, and cartridges on this machine. The ability to do inert headspace and many other capabilities, and it's available for use on commercial lines. The other thing is with these systems, because they're a standardized design, they're much more reliable, but they're also reliable in many senses because in terms of the manufacturing, we're making the same system over and over and over again. We've gotten very, very good at it. There's no surprises in the manufacturing process. We have a robust supply chain, and you're going to get the product or the work cell when you need it. So it's delivered on time. It can be installed very quickly. There's no assembly on site required. And then the qualification is a straightforward process. Everything has been tested as a standard process, and you're more likely to be able to complete it relatively quickly. Same thing goes for operation. They use minimal number of people. They're not going to be in the same grade of clean room. You're going to be able to be installed in ISO 8 or grade D in Europe. And our support is also much easier to do. We always have spare parts on hand because we're always making the same system. And we always are able to support it because we have a common database of troubleshooting and best practices. And if we look at flexibility, as I mentioned, you have one machine, but we purposely call them work cells because they're flexible in what they can do. So, looking at the SA25 as an example, because it can fill so many formats, you can fill whatever your client is bringing you. And there's short decontamination cycles. Obviously, that helps with rapid changeover. But the ability to be very, very flexible actually allows you to do some things that you wouldn't be able to do with normal filling machines during clinical production. For example, because it has the ability to fill syringes, your customers can switch from vials to syringes in phase 2 or 3 and launch with the preferred final product format. The other thing that has been used in the past is the ability to, instead of using vials when you're doing your therapeutic dose determination, you can actually load pre-filled syringes with different dosages and make smaller mini batches to supply your clinical trials and save your valuable clinical trial material. So there's a number of ways that the inbuilt flexibility can help in contract manufacturing. And of course, the risk is reduced. It is a completely robotic filling process. There are no glove ports. The need for human intervention has been removed. And that's been removed by redesigning the process to make use of the ready-to-use containers. So there's a common handling interface because they're all supplied in the same configuration. Now, if there is a problem, there rarely is because most of the issues have been designed out, there are contingency plans in place. The system can be breached, the problem can be fixed, and then it can be decontaminated and the batch proceeds. And so this becomes a very reliable process with probably the lowest contamination risk in the industry. Recently, 8 of our users presented their production data, with a focus on environmental monitoring data in a paper by AAPS Pharm Sci Tech, which I'll hopefully be able to provide the link to. And it showed that the risk was an order of magnitude lower than with conventional isolated systems. And this gives you an example of what they look like. You can see their standard systems. We've got systems at CDMOs all over the world, at Emergent in Canada, at Wuxi in China, at Eurofins down in California, and at Fujifilm down in Texas. And all of these are active members of our user group. So our user group is a collaborative group that gets together to share best practices. And allows people to, basically learn from each other. So when we look at what the benefits are of adding aseptic filling to a CDMO business, one thing to realize, it's an anchor service. It helps retain customers and allows you to offer additional complementary services as well. The Cytiva aseptic filling work cells in particular allow you to add capacity quickly, and they remove operational risks because they're a standard product. They also allow you to fill any format that your client might need and switch quickly to other formats. And most importantly, they provide the highest level of assurance that a patient is receiving a safe product. And with that, Alex, I'll take any questions that the audience might have. I see that we do have some questions. And so I'll answer the first one. And the question is, have you already installed these machines in Class D, not in Class C? The answer to this, I believe we have installed a system in Europe in grade D. In the United States, they tend to go with the ISO 8 classification. One of the things is that very often what happens is the room is classified as grade C, not because of the requirements of the fill isolator, but because they're doing operations such as formulation that may require a grade C background. So yes, it can be installed in grade D, but be careful of whether or not there's other activities that might need to be performed there. And we look at the question number 2. And the question is there a way to insert microbiological plates inside the machine? The answer is no, there isn't, but viable environmental monitoring is performed in the case of the SA25 by active air sampling. In the case of the microcell, there's an alternate media-based passive sampling method used, but it's not a settle plate, but it's very similar. Then I have a question, how do you insert the consumables, single-use containers, etc., into the machine? And for this one, I'll go over first the flow paths. So the flow paths are provided as a completely sealed, pre-sterilized unit. They can be installed when the system is open to the clean room. And so in that case, they're installed in the pump. The pump is outside the system. There's a flange that can be sealed with a triclover that goes into the system. And then there's a sheath over the needle that's then placed in a holder. The system is then closed and it goes through a vapor phase hydrogen peroxide decontamination process. There is protection of the interior of the flow path from the exposure to hydrogen peroxide. And then when it's done, the tooling or the robots will take that sheath needle out of its container and start the filling process. For the ready-to-use containers on the microcell, they are placed again in the system when the door is opened, taken out of the bags in the clean room and put inside, and then their external surfaces are decontaminated by the same VHP process. For the SA25, there is an airlocked material transfer chamber where 32 tubs of materials can be placed on a carousel, and that again will go through a VHP decontamination process, and then they'll be brought in through that airlock one by one into the rest of the SA25 where they do the filling and the capping. The next question I have Is the system able to be used for BSL-2 activities, filling of active virus and bacteria? Can it be gassed by VHP or similar? The answer is yes. We have several customers that are making viral therapeutics as well as viral vectors. And in addition to doing the VHP decontamination at the start, they'll also do a post-filled VHP decontamination of the material before it comes back out of the system. We have another question. What about changing the formats of an existing machine later on? Let's say in a year or so. Is that time and cost intensive to change the software? Because CDMO probably don't know what comes in a year. This is a good question. And in fact, we solve that problem by most of our customers for the SA25 get multiple formats with their system. The tooling for vials can usually handle a wide variety of vial sizes, and they'll also get tooling for a syringe format. And as well, with some syringe formats, the 1 mil standard, for example, can also fill the 2¼ mil and the 3 mil. So there's that ability to already have those tooling present. If you need to install a new container format that's not covered by your initial group, it's a relatively short process to add the new format tooling, which is generally the addition of only a few additional pieces, in some cases none. I have a question from next question. Can you confirm again the time it takes to process a 2,000 unit batch in an SA25? Now that is going to depend on the container size, because there's a standard processing time for nest or tub of containers, typically around 4 minutes. So for the smaller containers where you can fit 100 in a nest, then you're going to be looking at producing 20,000 units within basically a double shift or a longer filling day. For the larger units, which may only have 12 per nest, it's likely that you'd have to do a smaller batch, and you wouldn't be able to do that in a filling day. Typically about 5,000. Okay, I have a question. What if the system is not reliable as expected? Can a glove be implemented on a hotspot later on to allow more flexibility? The answer is no. And the system, we've shown it to be reliable. We have an install base of close to, actually, I think over 50 work cells now. And the need for operator interventions has been almost non-existent. So in the case where there's a problem that does require an operator to clear a problem, breaching the system and then re-decontaminating does allow them to address that solution. And that's a process that, again, has not been done in production, but almost all of our users will model that in their media fill simulations. What about, next question is, what about BSL-3 products or OEB 5? That is something where, for those products, theoretically possible. It's obviously advisable to do a detailed risk assessment that is particular to those products, looking at all the risks. But it is a positive pressurized isolator with a very, very low leak rate that's tested at the beginning of every batch. Everything used is pre-sterilized and ready to use, and it can be completely sprayed down with CIP after a batch, and there are also safe change exhaust filters if that's required. But again, for those cases, you're going to want to do a detailed risk assessment. The next question, isn't the HP, and that's hydrogen peroxide, absorbed by the filling kit and then released in the product? And the answer to that is no. We have a protective sheath over the actual filling line, and that prevents ingress of the hydrogen peroxide during the decontamination. The next question is, can we install the single-use system, filling bag with tubing and needles, without gloves? And the answer is yes. That is installed by the operator in their clean room garb, which of course will include gloves on their hands, prior to decontamination. So it takes less than 5 minutes. It's a relatively simple process, requires no tools, can be done very, very quickly.
So thank you, John, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with the direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcast. Look for those announcements in your inbox. Goodbye.
Thank you. Goodbye.